So good afternoon, judges, colleagues, and members of the committee. I would like to thank the committee, first of all, for the opportunity to present our study, which looks at the effects of DUOX gene knockout in mice on Beclin-1 mRNA expression as a marker for autophagosome formation in gastrocnemius muscle. So autophagy, as the name suggests, is the process of cell degradation in order for self-preservation within a cell. In states of physiological stress, such as nutrient starvation, oxidative stress, or changes in hormone levels, autophagy is triggered. Several studies have found that the application of thyroid hormone has a stimulatory effect on autophagy, and this interaction proved beneficial towards muscle health. Therefore, in a state of excess or deprivation of thyroid hormone, there may be a link between the pathological pathways that occur in such a disease and autophagy activity. In our study, the DUOX gene is modified in mice to mimic different states of hypothyroidism and finally autophagy activity. We can see this line of thought in the introduction section. The DUOX gene encodes DUOX or dual oxidase, which are crucial transmembrane proteins that produce hydrogen peroxide. Hydrogen peroxide is then utilized in the iodination and coupling of thyroglobulin molecules and the subsequent formation of thyroid hormones. This will then activate autophagy proteins for autophagosome formation, one important protein of which is Beclin-1. Therefore, Beclin-1 formation will reflect autophagy activity. To test this, we can quantify Beclin-1 by the use of quantitative reverse transcription PCR, which detects mRNA Beclin-1 levels. For our cooperation with Gunma University, Japan, we were able to utilize different variations of mice according to their sex, either male or female, age, either postnatally 2 or 3 days, postnatally 15 days, or postnatally 25 days old, and phenotype, either with a homozygous DUOX gene knockout, a heterozygous DUOX gene knockout, or a wild-type phenotype. The phenotypes used were previously studied, and interestingly, it was shown that specifically the homozygous phenotype had significantly smaller physical characteristics compared to the other phenotypes. We used 3 mice of the same age, sex, and phenotype for each group. Then we were then able to dissect and collect samples of the gastrocnemius muscle for PCR testing. We utilized Sensipass SYBR-NO-ROX one-step kit with specific primers for Beclin-1 and beta-actin, which can be seen in Table 1. We were then able to analyze the data using ANOVA and Tukey test. For the results, the first graph represents the average and standard of mean error, each combination of age, sex, and phenotype by their mRNA expression levels, with the star is used to identify a statistically significant difference between all 3 phenotypes or just one compared to the others. Just from the eye test, we may see that overall, the knockout phenotype, regardless of age or sex, has the highest overall autophagy activity and is mostly significantly different, therefore contradicting our hypothesis. This next graph displays how sex differences affect autophagosome formation as aging progresses. Interestingly, we see a similar trend of a drop in autophagy activity at 15 days postnatally and higher autophagy activity mostly at 2 days and also 25 days postnatally. Through these results, we found that the homozygous knockout mice phenotype were able to generate a higher level of autophagy activity, which may indicate a compensatory state of increased autophagy. The drop in autophagy activity in all 15 postnatal day mice may be attributed to the early weaning stage of mice and how this may disrupt autophagy, while the effects of female sex hormones as opposed to male sex hormones produce a higher level of autophagy activity, indicating a complex interplay between puberty, thyroid disease, and autophagy compensation that needs further research upon. And in conclusion, we have made the discovery that the correlation between DUOX levels and autophagosome formation does exist, but more importantly, it is found that there was a compensatory increase in autophagy during significantly lower thyroid hormone levels. Specifically, we found that in mice, females have a stronger autophagy response than males, with the exception of P25 males and a stronger response in the homozygous knockout phenotype. Further studies are required to uncover the specific pathways. However, our study has uncovered the general relationships and links occurring. Thank you all very much for this opportunity.
Now you have 3 minutes for the questions and answer session.
So your study, you are using the knockout genes, right? In the mice.
Yes, very true.
So my question is about how can you sure about your test mouse that already has some knockout in these genes? How do you confirm that?
Okay, so The confirmation is actually done by the cooperation part with Gunma University. So we, as Universitas Padjadjaran, is more so to the extraction and also the analysis part using PCR. But however, for the testing, it's usually done by PCR testing, looking at the specific gene where they have inserted a specific marker, which the details are quite unclear for me, but we have entrusted the Gunma University Japan for the sampling as to my knowledge, it's actually the neomycin-resistant gene, which is used for being able to detect if there's a knockout in the gene.
Thank you.
Any more questions for our judges?
My question is, rather than using gastrocnemius muscle, can we use other tissue?
Yes. So in this study, we have used gastrocnemius muscle and also the soleus muscle because these muscles are mostly utilized in mice and they're very active metabolically. And so we hope that by using these mice, it's going to be able to help us get a clearer picture on the autophagy process or the clear picture on autophagy for the use of gastrocnemius and in our case, it's soleus muscle.
Both gastrocnemius and soleus muscle are skeletal muscle.
Yes,
Right.
Skeletal muscles.
Did you try or you have any idea you're gonna have the same result if you use other kind of tissue like hard tissue or smooth muscle?
Oh, yes. So our research is actually part of a certain chain of research, and there are other studies that we have done on liver tissue, where we look at not the Becklin-1 protein, but instead the LC3 protein. There's different types of autophagy proteins that we like to compare using not just PCR, but also Western blotting to further confirm our hypothesis. However, this one is the one that I present today because it also represents the findings that we found in all the other studies. It's pretty linked together.
Okay, thank you so much.
May I ask questions?
Yes.
Hello. Yes. Do these mice, the knockout mice, survive until they age or mature, which implies that these genes might not be as lethal as we expected?
Yes. So in terms of how it links between the DUOX knockout and also how the survival rate of these mice, it has been previously researched upon. On one hand, it is seen that yes, the postnatal mice that are 25 days old don't really live longer than that point, in which why we also use the postnatal day 25 mice because it is said to be on the early stage of puberty, while they're also still they haven't generally they haven't died on their own yet, and that's why we use these 3 stages to represent the early neonatal stage, the early weaning stage, and also the early pubertal stage, where they are still viable, and all 3 are extracted after being euthanized and not by their own passing.
Okay, thank you so much.